Good morning, class 10. Welcome to online bio classes. Today we'll continue with the same chapter number 4, absorption by roots. In the previous class, we had already studied about the different methods like definition of osmosis, definition of diffusion, the differences between osmosis and diffusion. Today we'll study about different types of solutions. You are already aware that solution is a mixture of solute and solvent. Solute is the substance which dissolves, like sugar is the solute, and the substance in which a solid dissolves is known as solvent. Like water is the solvent in which most of the materials or substances will dissolve. Now, according to the nature of solute, three different types of solutions are there. What are they? Number one is called hypotonic solution, number two hypertonic solution, and third one is isotonic solution. So let's start with the first, isotonic solution. What is isotonic solution? A solution in which the contents of the solute is equal both inside the cell and outside the cell. That is, equal amount of water is present on both inside the cell as well as outside the cell. So if the concentration of water molecules on either side of the cell membrane is present in a plant cell or animal cell, it is equal, there will be no osmotic pressure, there will be no net movement of water molecules from one part to the other. So such a solution is called isotonic solution. So what is isotonic solution? Already I said. Now next one, number two is hypotonic solution. Hypo means less. Okay? Tonic means the tonicity represents the strength of the solution. The prefix hypo means less. Hyper means more. So this way you can learn. Hypotonic solution is such a solution in which the concentration of solvent molecules is more inside, means hypo means it is less outside the cell and it is more inside the cell. So I will say like this. Suppose this way you can understand, a beaker containing water. I have kept a cell. If I keep a cell, suppose RBC, inside a hypotonic solution, this one is hypotonic. Okay? Hypotonic solution. If I have kept RBC there, red blood cell, in a hypotonic solution, what will happen? Hypo means already containing less water, means more of solute is there, isn't it? More of solute is there? No. More of solvent is there, that is water is more, solute is less. So hypotonic means more of solvent is there, water is more, and the solute is less. So it means as compared to this hypotonic solution, the RBC contains less water, less solvent, and more of solute. So water molecules will diffuse into the cell and it will cause swelling or bursting of RBC. Okay, the RBC will swell. Now if the same RBC is kept in a hypertonic solution, like this is hypertonic solution is there. Hyper means more, extra, okay? Tonic means tonic represents the strength of the solution. So if a solution contains more of solute, it represents the strength of the solid. Like more solute is there, less of water is there. So as a result, the water molecules which are more inside the RBC, it will come outside by exosmosis. The process is called X means coming outside the cell. And here it is known as endosmosis. Endosmosis. Okay? Endo means the water molecules going inside the RBC. And exosmosis means here the water molecules leaving the cell and going to the surroundings. That is in the outside. So third type of solution is hyper. Only this spelling you have to check. Hypotonic solution. So what is hypotonic solution? How to define it? Hypotonic solution is if the concentration of water molecules outside the cell is less. So here water is less water in hypertonic solution. Here water is more. That will help you to understand, okay? More water. In the hypotonic solution, there is more water. In hypertonic solution, there is less water. Means solid is more. So that's why it is called hypertonic, okay? There is less water means more solid, and here it is less solid. So according to the concentration of solid, it is called hypotonic. And here in case of isotonic, as we had already discussed, the amount of solid is equal. Means solvent and solid both are equal. Whatever is the amount of solvent, that is water molecules present inside the cell, it is equal to the amount of solvent or the water molecules present outside the cell. So hope you have understood all the three types of solutions. Isotonic solution is such a solution in which the concentration of solvent or water molecules, we can define it in terms of solvent also, solid also. Okay? So if the concentration of water molecules on either side, suppose this is a cell membrane, okay, cell membrane has tiny pores in it, you know that. If water molecules like 1, 2, 3, 4 water molecules, here also 1, 2, 3, 4 water molecules, these are represented as water molecules. If they are equal, 4, 4 on either side, such a solution is called isotonic solution, because there will be no net movement of water molecules from side A to side B, that is from outside to inside the cell membrane. Neither the water molecules will move from inside the cell membrane to the outside. As here you can see, endosmosis occurs, means water molecules are going from outside to inside the cell, and here water molecules are coming outside the cell. This causes shrinkage of RBC, that is red blood cells will shrink because of loss of water. They will lose their turgidity or the hardness, and that will cause shrinkage of the protoplasm, which is known as Plasmod Lysis. So that term also you have to learn what is Plasmod Lysis. But one by one we will do it now. First let's complete this one. Have you understood endosmosis and exosmosis? Exosmosis. Check the spelling, okay? Exosmosis spelling is EXO. Exo means coming outside. ENDO. Endo means going inside. Got it? Exo means coming outside. Endo means going inside. So, what is endosmosis? Endosmosis, as the word you know, osmosis is the movement of solvent or water molecules from higher concentration to lower concentration through a semi-permeable membrane. A membrane is required in case of osmosis. So here it occurs from outside to inside the membrane, inside the cell. So type of movement of water molecules from outside to inside the cell is called endosmosis. And exosmosis is the movement of water molecules from the cell to the outside. Okay? Means from inside the cell, the water molecules move out. As you can see in the arrow I have indicated like this, water molecules coming outside. Okay, so that is called exosmosis. Now endosmosis, second difference you can write, it occurs in hypotonic solution, whereas exosmosis occurs in hypertonic solution. Got it? Third difference you can write, endosmosis, because water molecule is going inside, it results in the swelling of the cell. The size of the cell will increase because water molecules will fill the vacuoles, that is the empty space which are present in the cell, and cause the enlargement of the cell, that is the size of the cell will increase. Whereas exosmosis is the loss of water from the cell, that will cause the shrinkage of the size of the cell, or the, we can say it will result in shrinkage. Yeah, that is the best word. The size of the cell will reduce. Okay. Now next is swelling up of raisins. One example is given in your book. Raisins you see, in what in Hindi it is called kismis. Actually it is made from the grapes which are dried. So grapes, as you know, they are swelling like this. When the grapes you buy from the market is swollen like a ball, spherical in shape. But when the same grapes are subjected to drying, means when we dry them, then they will be plasmolized, or they will lose their water molecules by exosmosis, and they will result in the formation of raisins or the kismis. Now once you are making some sweet dish like kheer and all, you put those raisins inside it. Then what happens? Due to the process of endosmosis, water molecules will enter inside those raisins, and they will result in the swelling. So raisins will swell like grapes, isn't it? And that causes the swelling of raisins. So swelling of raisins or the kismis occurs in endosmosis, that is hypotonic solution is called endosmosis, whereas the drying of grapes due to loss of water from it is called exosmosis. Okay? And it results in the formation of raisins. Now next is osmosis and the plant cells. Osmosis in plant cells occurs through the semi-permeable membrane. A plant cell is in pure water. It takes water by the process of osmosis through its selectively permeable membrane. And you know that this selectively permeable membrane is present in all the plant cells because cell membrane is there which is selectively permeable. Now what is the importance of osmosis for the plant cells? How osmosis is good for the plant? How it is beneficial for the plant? Number one, osmosis results in the turgidity. It helps in the movement or it helps in maintaining the structure of different plant parts. Like because of osmosis, water molecules will enter inside the plant cell and that will help to maintain the structure of leaves, stems, roots. Okay. Number two, the movement of water molecules from one cell to the other, it occurs by the process of osmosis. And third one is absorption of water. You know that roots are present in the soil. So plants will absorb water from the soil through their roots by the process of osmosis. So what are the three uses? Absorption of water, maintaining shape and size of different organs due to turgidity which is provided by osmosis, and the movement of water molecules from one cell, plant cell to the other occurs by osmosis. Now next is osmotic pressure, a very important topic. What is osmotic pressure? And you have to understand it also because osmotic pressure means you have studied in physics about the pressure, no? Force per unit area is called pressure. But here also it applies in a different way. You can see what is the difference between osmotic pressure and osmosis. Okay. What is osmotic pressure? In simple words I will say, the pressure which stops the process of osmosis is called osmotic pressure. Okay, in your book the definition is given. The force exerted means applied by, the cell sap or solution on the water molecules to let it move from outside to inside the cell through a semi-permeable membrane is called osmotic pressure. So very nice definition is given. What is osmotic pressure? The force exerted by the cell sap. What is that force and what is cell sap on the water molecules? You know that a plant cell contains a big vacuum or empty space which is filled with water and minerals. Those water and minerals are called sap. This one is the vacuole. Okay? V-A-C-U-O-L-E, vacuole. Vacuole is the empty space which is present inside a cell. And they are present in very large size inside the plant cell. In animal cells, the vacuoles are small or almost negligible. And these vacuoles has a covering also. That membrane is called tonoplast. Okay? We have to learn the name of the... And inside the vacuole, what is present inside? That is known as cell sap. I will write the complete word, cell sap. Cell sap consists of water plus minerals are also there. Different types of minerals, you know. Water and minerals are present inside this cell sap. Okay. Now, what is osmotic pressure? The force exerted by the concentrated cell sap. What is the meaning of concentrated cell sap? A cell sap which contains more solid, that is more of minerals in it. Water is already there. If minerals are more, it becomes concentrated. If minerals are less in quantity, it becomes dilute cell sap. So the force exerted by concentrated cell sap on the water molecules to let it move from outside to inside the cell through the semi-permeable membrane. This one is cell membrane and the cell membrane is semi-permeable. the cell membrane is SPM. SPM means semi-permeable membrane. Okay. Why? Because it will allow only selected particles to pass through it. So osmotic pressure is directly proportional to the number of solid particles in a given amount of solvent. If the solid particles are more, then more will be the osmotic pressure. So if the solid particles are less, less will be the osmotic pressure. So whenever I use the word what is osmotic pressure, you can understand the pressure applied by the solid particles which are present in that solution. Okay, got it? Now, one diagram is there in your book to explain the process of osmotic pressure, in which experimental setup has been shown, in which solutions are kept, like here solution A is there, solution number A, here solution number B is there. In solution A, what is kept is, it is the water molecules. So these are the water molecules which is kept in solution A, whereas in solution B we have the sugar solution. Sugar solution is kept in B. Okay. Now you can see that water molecules are more in A, less in B side. So water molecules will move from A to B through the tiny pores which are present in the semi-permeable membrane until the concentration is equal on both sides. Okay. Now what is osmotic pressure? If we apply what we say that piston means a pressure valve will be there. Okay. So water molecules will diffuse from higher concentration to lower concentration. Here sugar solutions are there. Now I'll show it like this with a sugar solution. Now water molecules will keep on moving from higher to lower concentration. When we apply a pressure inside B, it will stop the movement of water molecules. That pressure which is applied to stop the process of osmosis, that is movement of water molecules, is known as osmotic pressure. Got it? So you can see that the pressure will resist the flow of water to the sugar solution. So pressure exerted to prevent the flow of water molecules from A to B to cross the semi-permeable membrane. This membrane is semi-permeable. Okay? From the higher concentration, because there is more water molecules inside A, only water is there, so of course it is more. Here it is sugar solution, means sugar particles are there along with the water molecules. Okay, because sugar is dissolved in water to make the sugar solution. So next is osmotic pressure is the minimum pressure. One definition is there, that must be exerted. Exerted means applied to prevent the passage of solvent solution. Solvent means the water molecules when the two are separated by a semi-permeable membrane. So whatever I told in the beginning, that simple definition, what is osmotic pressure? The pressure applied to stop the process of osmosis is called osmotic pressure. Okay? Now next is turgidity, the next topic we have to do. But before that, one exercise, section exercises are there. Hope you can do it yourself also. Like first question is, what is diffusion? Diffusion is the movement of molecules of a substance from higher to lower concentration. Then what is its importance? The importance of diffusion is it helps in the movement of molecules of liquid, gases from one place to the other, from higher concentration to the lower concentration. Like diffusion of gases occurs inside the water, which helps the aquatic organisms to survive. Diffusion of water vapor from the surface of leaves during transpiration helps in the cooling of plant. Understood? Now next is, how does diffusion occurs in plants? Why does carbon dioxide diffuse into the leaves? You know that why carbon dioxide plants need? To make their food by photosynthesis. Then can osmosis be regarded as a diffusion? Yeah, we can say osmosis is diffusion. Give reasons, because it is the movement of molecules from higher to lower concentration. Okay? But here the semi-permeable membrane is also there. Why do raisins swell in water? Just now I explained. Under what osmotic condition does water not enter a cell? Water will not enter the cell in isotonic solution. Okay? when the osmotic condition is asked, when the osmotic pressure is applied. Then name the process or solution whose concentration is equal to that of cell sap. Such a solution is called isotonic solution. A solution whose concentration is greater than that of cell sap, it is called, greater means hypertonic solution. A solution whose concentration is lower than that of cell sap, it is called hypotonic solution. Two solutions having equal osmotic pressure, both the solutions are called isotonic solution. Then fill in the blanks there. The plasma membrane is semi-permeable or fully permeable. It is semi-permeable. Number 2. Dash is the membrane of transport of minerals in plants, cell wall or cell membrane. It is the cell membrane which helps in the transport of minerals. Osmosis is the diffusion of water across a partially permeable, not fully permeable. It should be partially permeable membrane. The process by which molecules distribute themselves within the space they occupy is called diffusion, not osmosis. Okay. Now next we have turgidity. What is turgidity? Turgidity means, wait, I will explain it nicely so that you can understand what is turgid, what is turgidity, what is the difference between them. Turgid means hardness. So when a plant cell is kept in pure water, it takes water through the cell membrane by the process of endosmosis. I had explained already, like this is the plant cell I have kept here, and this is the water molecule. The water molecules will enter into the plant cell by the process of, this is water, H2O, this one is the plant cell. So water molecules will enter inside the plant cell, and that will result in filling of water inside the vacuole. That will cause hardness of the plant cell. This condition is called turgid condition. T-U-R-G-I-D. Turgid means when fully distended. The outward, now some words are there, you must be careful about it. What is turgid pressure? Two words I will say, turgid pressure. The second one is wall pressure. What is turgid pressure? First, okay. Turgid pressure means the pressure which is applied by the cell contents. I'll draw it here so that you can understand. Suppose this is the cell, plant cell. Inside the cell we have the cytoplasm, vacuole and all the cell organelles. So pressure which is applied by the cell contents on the cell wall. This outward pressure it is. It is known as turgid pressure, TP. So what is turgid pressure? The pressure exerted on the cell wall by the cell sap, because if it is filled inside, it will apply pressure on the boundary, that is on the cell wall. So turgid pressure is pressure applied on the cell wall by the cell contents or the cell sap. Similarly, in order to resist or to stop this turgid pressure, if turgid pressure keeps on increasing, what will happen? If lot of pressure is applied by the cell contents, the wall may rupture or break, and it will cause bursting of the cell. In order to stop or resist this pressure from bursting of cell, the cell wall also apply pressure on it to counter this pressure on the cell contents. So wall pressure is the pressure applied by the cell wall on the cell contents or the cell sap. Okay, got it now? So what is turgidity? The plant cell in this state is called turgid. Like when the cell contents are applying pressure on the cell wall, then the plant cell is fully distended. You can see fully distended. Why? Because the cell contents are applying pressure on it, fully distended. This condition is called, the state is called turgid, and the condition is called turgidity. So how to define turgidity? Turgidity is the plant cell in this state is turgid, and the condition is called turgidity. So turgidity is a condition of a plant cell when the cell contents or the cell sap apply pressure on the cell wall. Okay, and it is fully distended. So plant cell has very strong cell wall, and the cell wall reacts to this turgid pressure and try to make it stabilize. That pressure which is applied by the cell wall to counter the turgid pressure is called wall pressure. Got it now? Now what is the importance of turgidity? How turgidity is beneficial to the plant? Because it helps to maintain the shape of the plant cell. It gives the shape of the soft organs. The leaves and flowers will quickly droop down and become flaccid. It will become soft and flexible when there is a scarcity of water. So when the water molecules go inside the leaves become turgid and fully distended. Number 2, it helps in the growth of roots. It is the turgid pressure or the turgidity which helps the roots to enter the hard surface of the soil. And that's how the roots are able to grow deep inside the hard soil. Turgid pressure helps in the opening and closing of stomata. That helps in the exchange of gases. In some plants, the movement of leaves or the plant organs are caused due to change in turgid pressure. You have seen touch-me-not plant. Scientific name is Mimosa pudica. Once you touch or even by the strong wind, the leaves of touch-me-not plant will close. Why? Because they lose their turgidity. And 1 second after some time, the water molecules will enter inside the leaves and they will maintain their turgidity and the leaves will once again open. Now next condition is flaccidity. Just opposite of turgidity. Flaccidity. What is flaccidity? Just now we read that turgidity is maintaining the shape of the cell. Flaccidity is the loss of turgidity, which helps to shrink the cell, or it helps to deform the shape of the cell. So in your book the definition is given. When a plant cell is placed, like this is the plant cell, if this solution is hypertonic solution ETL. If a plant cell is kept in a hypertonic solution, then what will happen? Water molecules will move outward. Okay? I will draw it here. This is a hypertonic solution. A plant cell is kept here. Hypertonic solution. The cell will lose water. The water molecules will come outside. Why? Because there is more water inside the cell, less in the surrounding. Because it is hypertonic solution, so water molecules will move outside and the cell will now shrink. You can see the condition of the cell now, like this it will become. So this shrinkage of the cell or the cytoplasm of the cell due to loss of water is called by loss of water by which process? Exosmosis is called flaccidity. The state of the cell when the cell will shrink like this, it is called flaccid state, and the condition of the cell is called flaccidity. So what is flaccidity? Flaccidity is changes in the shape of the cell due to loss of water from the cell by exosmosis when it is kept in a hypertonic solution. Hope you have understood. Got it? Okay. Now next, imbibation. What is imbibation? We have seen that during the rainy season now only, we have plenty of rains. The wooden doors and windows, they will absorb water from the atmosphere and they will become tilted. And it's hard to close the doors and windows. So this movement of water molecules inside the wooden doors and windows, which is made up of cellulose, and you know that cellulose are hydrophilic, that is, they have a strong affinity for water, which results in deforming the shape of wooden doors and windows. It causes the movement of water, and that results in difficulty in closing wooden doors and windows. So imbibation is the phenomena or the process in which living or dead cells of the plant in their dry or semi-dry state absorb water by surface attraction, which is created by the hygroscopic substances like cellulose, okay, hemicellulose and pectin materials. So what is imbibation? Imbibation is the absorption of water by surface attraction into the wooden objects. Okay. Now next, the swollen seeds. One more example I will give of imbibation. You may have seen that the seeds, when they start germinating, first of all they will absorb water from the soil and they will swell, and that will result in making the seed coat soft, which will help in the growth of radicle and plumule that will result in the formation of root system and the shoot system. Okay, so next we have to do is Plasmod Lysis. Okay, the next topic which we have to do is Plasmod Lysis. Plasmod Lysis. As you can see, there are two parts, plasmo and lysis means breaking. So actually there is no breaking here. Breaking of cell membrane may occur when it will shrink towards the center. Plasma means plasma. So plasmolysis, what is the definition? The shrinkage of protoplasm. Shrinkage of protoplasm of a cell in a hypertonic solution. In a hypertonic solution is called plasmolysis. So when the plasmolysis starts, the beginning of plasmolysis is called incipient plasmolysis. So if it is asked what is incipient plasmolysis, in your book also definition is given, INCIPINT. Incipient plasmolysis means the start of plasmolysis, or the beginning is start of plasmolysis, okay, or the beginning stage, the initial stage of plasmolysis, okay, is called incipient plasmolysis. Whereas when the cell is showing incipient plasmolysis, now when the plasmolysis occurs, what are the conditions required for it? We need a hypertonic solution. So I will just show you one example how it happens. That if I keep a cell in a hypertonic solution, like this one is the cell wall, this is the cell membrane, okay. Now if I keep it inside a hypertonic solution, then what will happen? All the vacuoles are there which is filled with cell sash. The vacuoles will be drawn outward and there will be shrinkage of protoplasm, means leaving the cell wall will remain intact as it is. Remaining part, that is the cell membrane and all, this is the cell membrane, this is the cell wall. So the cell membrane will shrink and come towards the center. So you can see in the next diagram which I am drawing, the cell wall is as it is. This is the plant cell having the cell wall, and now the cell membrane is here and all the cellular contents. Since it is shrinking of the entire protoplasm towards the center of the cell. That process is called plasmolysis. Okay, and it occurs in hypertonic solution. And what is incipient plasmolysis? The initial or the starting stage of plasmolysis is called incipient plasmolysis. Now, if I keep a plasmolyzed cell in a hypotonic solution, like take this cell and keep it in a hypotonic solution. The solution is hypotonic. Now once again, in hypotonic solution, water molecules will enter inside the plasmolyzed cell and it will return back to the original condition. This condition is called deplasmolysis. So what is deplasmolysis? The reverse of plasmolysis, or the condition of regaining turgidity. Turgidity means hardness, is called deplasmolysis. Okay. Now next, we can say that plasmolysis is outflow of water, because water molecules are flowing outside. That's why the cell is shrinking, or it has shrinked. Outflow of water there. Whereas deplasmolysis is the inflow of water inside the cell. Okay. Now once a cell is fully plasmolyzed, means completely plasmolyzed, it will result in the breaking of cell membrane away from the cell wall. So now if we try to put it in a hypotonic solution also, like if exosmosis continues, this is exosmosis, movement of water molecules, it causes permanent plasmolysis, and a permanent plasmolyzed cell cannot return back to the original state. Why? Because of the breaking of cell membrane away from the cell wall, which causes damage to the and that's why the word is used. Plasmod Lysis means breakdown. Okay. So if permanent plasmolysis occurs, a cell cannot regain the turgidity even after it is transferred in a hypotonic solution. Now, what is active transport? The next topic. You know, transport means movement from one place to the other. Active transport means movement of molecules from lower concentration to higher concentration, that is against the concentration gradient, by using energy in the form of ATP. Whereas passive transport, example diffusion and osmosis are examples of passive transport, in which there is movement of molecules from higher to lower concentration, that is along the concentration gradient. Okay, from higher to lower, not from lower to higher, without any expenditure of energy. So differences also you can learn, which is given on page 39. Then the next topic is root pressure. Root pressure is like due to continuous flow of water inside the root hair cells from one cell to the other, a pressure develops inside the cortical cells of the root. This pressure is called root pressure. So cortical cells of the root develops a pressure exerted by the cells of the cortex of the root upon their liquid content under a fully turgid condition, forcing the quantity of them to move into the xylem. You know that water molecules move in a systematic way, starting from the soil particles from where they are absorbed through the root hairs. The root hair is made up of different types of cells, like we have the outermost epidermal cells, which changes into root hair. Then from there it moves into these cortical cells. Then from cortical cells it enters into the xylem vessels, and through the xylem vessels the water molecules are transported to upper parts of the plant. So this pressure can be shown with the help of an experiment also in the lab. We have to take a stem, like a plant we have to take, potted plant. With the stem we have to cut this stem, okay, and then we have to hit a glass tube, and we have to connect this glass tube with a rubber, just like LPG gas cylinder rubber tube is here, and this one should be connected with, okay, the experimental part which is shown. This one is connected, the rubber tube is connected with a manometer. Manometer is like, which is filled with mercury. You can see the diagram is there in your book. Mercury filled column is there. Okay, it is filled with mercury, and it is fixed with the help of a stand. Stand is here. Now, what you will see in this experiment, demonstration of root pressure. You will find that this potted plant is here. This contains soil and the water. The water molecules will move up into through the roots into the stem. This is the stem of the plant, and then it will enter. The water molecules will enter inside this glass tube, from the glass tube into the rubber tube, and then it is filled with mercury. The mercury column will rise, and once the mercury column will rise, why? Due to the root pressure which is shown by the plant here, because it will force the water molecules to move upward, and that can be shown with, or that can be proved with the help of increase in the level of mercury inside that manometer. So mercury level will rise. Okay. Now last topic is ascent of sap. You know what is ascent? Ascent means movement. So movement of water and minerals from the roots to the upper parts of the plant is called ascent of sap. We know that in lower plants like herbs and all, only the root pressure is sufficient to push the water molecules to the leaves. But in higher plants like eucalyptus and all, this process is very complicated because it involves many more processes. Like number one, root pressure, already you know. The root pressure developed in the xylem vessels helps to push the water molecules to the leaves and the branches. Then capillary force. What is capillary force? Xylem vessels and tracheids, they are in the form of thin capillaries or tubes. So when water enters inside the thin tube, it has more pressure. The xylem capillaries exert a capillary force, so which will help in the ascent of sap. You can think it like this with the help of the example of the pipe system in your home. the more thin the pipe is, higher is the pressure applied on it. If you take a pipe with a bigger diameter, the pressure will be reduced. Like if you take 3/4 pipe, more pressure will be there. If you take 1-inch pipe, the pressure will decrease. So capillary means the thinness of the tube will increase the force with which water travels. Then comes cohesion and adhesion of water molecules. Dixon and Joly proposed that a strong cohesive force means the water molecules will stick to each other and form a continuous column of water. You have never seen that drops of water do not travel in the form of separate units. They travel in the form of a column, a complete continuous column. Not in the form of packets like this one water going again, another water is traveling, not like that, no. These water molecules stick to each other with the help of cohesive forces. Force of cohesion means the force which helps to attach the water molecules is called cohesion. And adhesion means since the water molecules are traveling inside the xylem vessels, the force which develops between the xylem vessels and the water molecules, it is called force of adhesion. Got it? So cohesive forces is between the water molecules which helps them to stick together, and adhesive forces are present between the water molecules and the xylem vessels. Now next one is transpirational pull. Due to loss of water from the leaves of the plant, we can say a vacuum is created in the leaves or empty space. That empty space is filled by water molecules which are drawn from the leaf xylem. The leaf xylem will draw water from the stem xylem, and stem xylem will draw it from the root xylem. So transpirational pull means due to the process of transpiration also, a suction force is developed in the roots which help to draw more and more water from the soil. Okay, so we have completed it. Today this much only. Next we have to do the exercises. I will discuss in the next class. So as homework, what you can do, you can try the objective questions of the exercises, and then one last topic is there which is there in your syllabus to demonstrate that water is conducted through xylem. So I will explain you in the next class along with the exercises which are left. I will try to say the answers of these exercises, and then we can start the new chapter. So after watching this video, you just write your name in the comments. Thank you.